Hello everyone, and welcome to this video about the Ramamoorthy Lab at the University of Michigan. I am Dr. Giacomo Di Mauro, a recent alumnus of the research group, class of 2021. If you're here, you probably are considering joining the group. So, to help you out, it will be my pleasure to briefly introduce some of the research I've done in the lab. The research I did in the Ramamoorthy group is on edge between biophysics, physical chemistry, and chemistry of materials. But that seems quite broad, isn't it? So let's try to understand more about it by discussing some of the details. Now, if you think about biophysics, you might think of the science that applies the law of physics to biological phenomena such as cellular mechanism, cell membrane behavior, protein structure, and protein dynamics. If you think of physical chemistry, you might connect it to spectroscopy, the Schrödinger equation, the harmonic oscillator, and many more equations. Finally, what about chemistry of materials? It is usually associated with materials such as metals, semiconductors, ceramics, and polymers. But the real question is, how do these far-apart fields meet in the Ramamoorthy lab? To figure this out, it might be helpful to look at the biological systems from a different angle. For example, cell membranes are materials that separate cellular compartments from the rest of the universe. They are complex biological systems, and among their building blocks, there are lipids and membrane proteins. These last characters are crucial because of the variety of functions they play. In fact, membrane proteins are encoded in about 1/3 of the human genome and are targeted by about 60% of all drug targets. However, the lipid environment cannot be neglected and being considered simply as a passive barrier for cell compartments. Indeed, lipids can affect proper folding, assembly, and functions of membrane proteins. Therefore, since their presence is vital, we need to mimic the lipid environment to study membrane proteins. The importance of membrane proteins in many fundamental cellular events has driven a marked increase in the use of membrane mimetics for exploring these proteins and as therapeutic agents. A variety of membrane mimicking systems are available, and our lab has experience with most of them. But in this video, I will focus on nanodiscs, or nanoparticles made of a phospholipid bilayer patch surrounded by an amphipathic belt, or simply sushi-like nanomaterials. The main challenge associated with biophysical analysis of membrane proteins is the need for detergent extraction from the bilayer environment, which in many cases causes the protein to become insoluble, unstable, or display altered structure or activity. Recent technological advances have tried to limit the exposure of purified membrane protein to detergents. Nanodiscs have been helpful in providing structures of quite challenging proteins, and here are just two of the many examples available in the scientific literature. The AC3 cytochrome AA3 supercomplex and the ACRB are just two of them. However, the term nanodisc is quite general. It is in fact an umbrella term for an enormous variety of systems available. Inspired by the high-density lipoparticles, or HDL, historically, the first were presented in the early 2000s, and they had a tremendous impact in the field. In 2009, the discovery that a synthetic polymer named SMA could form similar nanoparticles shocked the field once again. Called SMA lipid particles, or simply SMALP, synthetic polymers have the remarkable feature of extracting membrane proteins from their native lipid environment in few steps without the use of detergents. And since then, the interest in these polymers have kept growing exponentially year after year. Early research was focused on elucidating the mechanism of formation of the SMALP, focusing on the ability to dissolve membrane bilayers based on their formulations, also focusing on the membrane mimicking behavior. Throughout the years, a variety of different formulations have been proposed, and in the Ramamoorthy lab, we played a significant role by introducing many of them between SMA and on-SMA-based polymers, as highlighted here. In the Ramamoorthy lab, we engineered a small molecular weight polymer and achieved significant new features such as size tunability, increased tolerance toward divalent cations, and lower pH values. This allowed many NMR studies involving 1D and 2D spectra on diamagnetic and paramagnetic samples. Moreover, we were also able to align them in the magnetic field, allowing even more complex studies in solid-state NMR. Thanks to the different electrostatic charges, we can investigate membrane proteins with different isoelectric points, ensuring that they will not interact with the polymer belt of the nanodiscs. Furthermore, the introduction of chelating groups allows the presence of paramagnetic metals on the outer belt of the nanodisk, offering a unique theory NMR approach to accelerate the data acquisition and increase the overall sensitivity on NMR spectroscopy. In conclusion, there is so much more research to be discovered in the Ramamoorthy lab, but to summarize, join the group if you're interested in NMR spectroscopy, a multidisciplinary environment, cell membrane biophysics, protein-protein interactions, and protein-membrane interactions. You need more? Please check out the website and do not hesitate to contact Prof. Ramamoorthy via email for more info. Besides, check for our publications on both Google Scholar and our website. That said, good luck, and I'm sure you'll make the right decision. Good luck and ciao ciao.